Okay, so today we're going to be demonstrating the different biochemical tests and media that can be used to identify some differences in bacteria that you're not able to differentiate via staining alone. So we've got 4 different tubes that we're going to use, and this is all in the Enterobacteriaceae lab. And we're going to start with the SIMS media. So the SIMS tube comes as a solid here, and that is a medium in a tube, so it's hard, it doesn't move. So what we're going to do is we're going to pick up some bacteria from a plate on an inoculating needle. So here's our inoculating needle, which she's going to flame, let cool, and then pick up some bacteria from the plate. What's unique about this is that this SIMS media the SIM stands for sulfide indole motility, and what he's going to do is stab that. So here he's picking up some bacteria, and he's going to stab that into
I'm going to come right back out, same exact stab line.
Yep, you go right in and right out. And he's gone down about maybe an inch or so. And let's see if we can see this. Not so much. But you'll be able to see when the bacteria grow. So the motility part of this, you're going to be able to see movement. If the bacteria has movement, it'll move away from the stab off to the sides, and you'll see those bacteria growing. If they don't have motility, they'll grow right around where the stab site is. The indole is going to come about because the SIM contains an amino acid tryptophan, and some bacteria can metabolize tryptophan and produce indole as a byproduct. The presence of indole is we're going to be able to demonstrate that by adding something called Kovacs reagent to the culture after we've grown it. And if indole is present, it's going to react with that reagent and produce a red color. If not, you won't have a color change. And then the sulfide comes about because some of the bacteria can produce hydrogen sulfide through metabolizing sulfur-containing amino acids. This media, this SIM, contains sulfur-containing amino acids plus metallic ions like iron, which are going to act as indicators when they combine with hydrogen sulfide, and you'll see a black precipitate if that bacteria is able to metabolize that. So the next one that we're going to do is TSI, or triple sugar iron.
This is Slant.
This is a Slant. So here's the TSI, triple sugar iron Slant. So what we're going to do this time is we're going to inoculate the Slant by stabbing the butt portion of the tube and then streaking the Slant part. So he's going to go in, he's flaming his tube, or his inoculating needle. He's going to let it cool. Did you pick up bacteria?
Nope, forgot that whole step. Sorry.
So he's flaming the neck. He's got bacteria on his inoculating needle. He's going to go in and stab down towards the bottom in the butt.
I'm going to stab right now. Go all the way down. I can see the needle. Pull it all the way out. And then I'm going to zigzag across the top here. Right. I'm going to show him the... Can you see it or no?
Kind of.
Kind of? Yeah, I can see it from here. Zigzag pattern. Okay.
Yeah. All right, so that's the TSI. So the next one we're going to look at is the MRVP broth. So this one is a broth. And
Metal red Welchbrown.
So in this one, we're heating up his loop. We've switched from the needle to the loop. And this test is going to indicate glucose fermentation. And you'll divide this broth up into two and do two different analyses on it. If glucose is present, you're going to see a color change that will lower, that changes to red if the pH is low, which indicates that glucose is the microbe is using glucose during fermentation. For the other test, the Voges Proxauer test, that's looking to see whether or not in the process of utilizing glucose for fermentation, if it also will utilize acetyl methylcarbinol. And again, for that, you would have a color change or no color change.
So, the Pseudomonas?
For this last one, we're looking at the Simmons citrate agar, and this agar starts off as this like greenish color, and it's a slant. We're not stabbing anything for this one. We are just going to do a regular streak up the slant, but we are using an inoculating needle because we want to take a tiny amount of bacteria from the plate, and so with using just the needle, we'll get less bacteria than we would with a loop. The reason for this is because using the inoculating needle will reduce the number of microbes we're transferring, and sometimes if you have too many microbes and they're dying, you can get a false positive result, which we don't want. So he's going in and just doing a regular streaking up that slant for this. So those are the biochemical tests that we're using for the Enterobacteriaceae lab. So we'll do those for all of our unknowns and then analyze the results when they're done.